Dalam industri 4.0, konektivitas kita semakin tak terbatas. Teknologi farmasi bergerak semakin maju, menghadapkan kita pada persaingan global yang mengharuskan kita untuk terus berinovasi. Lalu, bagaimana cara kita agar putra-putri bangsa mampu bersaing di era ini? Apakah kita mampu untuk menghadapinya? Semakin maju, menghadapkan kita pada persaingan global yang mengharuskan kita untuk terus berinovasi.
Fakultas Farmasi UMP merupakan fakultas farmasi tertua di Provinsi Jawa Tengah bagian Barat. Saat ini kami mengelola 3 program studi unggulan, yaitu Sarjana Farmasi, program profesi apoteker, dan S2 Magister Ilmu Farmasi dengan jumlah mahasiswa hampir 1.000 orang setiap tahunnya. Fakultas Farmasi UMP mempunyai visi untuk menjadi fakultas farmasi yang unggul, modern, dan juga islami. Kami berusaha keras untuk bisa berkolaborasi dan juga berperan aktif dalam pengembangan ilmu kefarmasian dengan institusi di dalam maupun di luar untuk menjadi fakultas farmasi yang unggul, modern, dan juga islami. Sebagian besar dosen di Fakultas Farmasi UMP mempunyai kualifikasi doktor yang merupakan lulusan dari universitas di luar negeri maupun universitas terkemuka di Indonesia. Alat-alat penelitian laboratorium yang dimiliki oleh Fakultas Farmasi UMP mendukung unggulan penelitian yang kami miliki. Beberapa penelitian unggulan prioritas di Fakultas Farmasi UMP berkaitan dengan biologi molekuler, pengembangan vaksin, pembuatan kosmetik yang aman dan halal, dan juga kebijakan kesehatan berdasarkan farmakoekonomi. Saat ini, Fakultas Farmasi UMP mempunyai kerjasama aktif dengan institusi di dalam maupun di luar negeri di bidang tridharma pendidikan tinggi, yaitu pendidikan, penelitian, dan juga pengabdian kepada masyarakat. Saat ini, Fakultas Farmasi UMP dengan keunggulan dan juga fasilitas yang kami miliki, Fakultas Farmasi UMP berharap untuk bisa berkontribusi lebih dalam penyelesaian masalah kesehatan di Indonesia. Selain itu, mahasiswa-mahasiswa farmasi juga kami harapkan untuk bisa berkontribusi secara real terhadap masalah yang ada di sekitar mereka. Selain itu, alumni-alumni kami bisa memberikan nilai tambah, bisa membawa nama besar Fakultas Farmasi UMP kepada masyarakat sekitar, sehingga secara umum Fakultas Farmasi UMP adalah fakultas farmasi yang unggul, modern, dan islami. Alumni-alumni kami bisa memberikan nilai tambah, bisa membawa nama
Kita
besar
mampu
Fakultas
mewujudkan
Farmasi
mimpi
UMP
putra-putri bangsa,
kepada masyarakat
melangkah
sekitar,
maju dan menjadi
sehingga secara
kebanggaan
umum
negeri.
Fakultas Farmasi
Mari
UMP
kita mulai
fakultas
perubahan pada
farmasi
diri
yang
kita
unggul,
untuk negeri
modern, dan
yang lebih
islami.
baik.
Alumni kami bisa memberikan nilai tambah, bisa
Kini,
membawa nama
kita
besar
mampu
Fakultas
mewujudkan
Farmasi
mimpi
UMP
putra-putri bangsa,
kepada masyarakat
melangkah
sekitar,
maju dan menjadi
sehingga secara
kebanggaan
umum
negeri.
Fakultas Farmasi
Mari
UMP
kita mulai
fakultas
perubahan pada
farmasi
diri
yang
kita
unggul,
untuk negeri
modern, dan
yang lebih
islami.
baik.
Alumni-alumni kami bisa memberikan nilai tambah, bisa
Kini,
membawa nama
kita
besar
mampu
Fakultas
mewujudkan
Farmasi
mimpi
UMP
putra-putri Dalam industri 4.0, konektivitas kita semakin tak terbatas. Teknologi farmasi bergerak semakin maju, menghadapkan kita pada persaingan global yang mengharuskan kita untuk terus berinovasi. Lalu, bagaimana cara kita agar putra-putri bangsa mampu bersaing di era ini? Apakah kita mampu untuk menghadapinya?
Fakultas Farmasi UMP merupakan fakultas farmasi tertua di Provinsi Jawa Tengah bagian Barat. Saat ini kami mengelola 3 program studi unggulan, yaitu Sarjana Farmasi, Program Profesi Apoteker, dan S2 Magister Ilmu Farmasi dengan jumlah mahasiswa hampir 1.000 orang setiap tahunnya. Fakultas Farmasi UMP mempunyai visi untuk menjadi fakultas farmasi yang unggul, modern, dan juga islami. Kami berusaha keras untuk bisa berkolaborasi dan juga berperan aktif dalam pengembangan ilmu kefarmasian dengan institusi di dalam maupun di luar negeri. Sebagian besar dosen di Fakultas Farmasi UMP mempunyai kualifikasi doktor yang merupakan lulusan dari universitas di luar negeri maupun universitas terkemuka di Indonesia. Alat-alat penelitian dan juga laboratorium yang dimiliki oleh Fakultas Farmasi UMP mendukung unggulan penelitian yang kami miliki. Beberapa penelitian unggulan yang menjadi prioritas di Fakultas Farmasi UMP berkaitan dengan biologi molekuler, pengembangan vaksin, pembuatan kosmetik yang aman dan halal, dan juga kebijakan kesehatan berdasarkan farmakoekonomi. Saat ini, Fakultas Farmasi UMP mempunyai kerjasama aktif dengan institusi di dalam maupun di luar negeri di bidang tridharma pendidikan tinggi, yaitu pendidikan, penelitian, dan juga pengabdian kepada masyarakat. Dengan keunggulan dan juga fasilitas yang kami miliki, Fakultas Farmasi UMP berharap untuk bisa berkontribusi lebih dalam penyelesaian masalah kesehatan di Indonesia. Selain itu, mahasiswa-mahasiswa farmasi juga kami harapkan untuk bisa berkontribusi secara real terhadap masalah yang ada di sekitar mereka. Selain itu, alumni-alumni kami bisa memberikan nilai tambah, bisa membawa nama besar Fakultas Farmasi UMP kepada masyarakat sekitar, sehingga secara umum Fakultas Farmasi UMP menjadi fakultas farmasi yang unggul, modern, dan islami.
Kini, kita mampu mewujudkan mimpi putra-putri bangsa, melangkah maju dan menjadi kebanggaan negeri. Mari kita mulai perubahan pada diri kita untuk negeri yang lebih baik.
Halo semuanya, silahkan dinyalakan videonya. Acara akan segera kita mulai. I also want to introduce Dr. Ika Yuni Astuti is the head of the department of the pharmaceutics. Yeah, and what is the name of the department? I'm always forgotten.
Pharmaceutical technology, Pak, Mr. Didik.
Yeah, so the department name is the pharmaceutical technology. So Dr. Ika is the head of the department. Tuh, ada Pak Zul. Gimana kabar Pak Zul? Ikut juga ini.
Ya Pak, Alhamdulillah.
Ya.
Ikutan Pak.
Ya Pak Zul, kalau mau berangkat itu ada beasiswa tuh dari Hungari, bisa tanya-tanya ke Bu Gita nanti.
Siap-siap Pak.
So Prof. Zubko and Prof. Piroska, apparently it is not only our student who are attending this lecture. Apparently our several staff from other universities who are usually work with us. For example, I saw Mr. Zulkifli from Makassar. He is from other island in Indonesia. Also, I saw several alumni from postgraduate program. There is Mas Angga so join.
Assalamualaikum Pak Didik.
Waalaikumsalam. Makasih Mas sudah join.
Ya, baik Pak Didik. Alhamdulillah.
Okay, yeah. Okay, then I will give it to who is the chairman, MC?
Arini the moderator.
Okay.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Welcome to the guest lecture of the Pharmacy Faculty, Universitas Muhammadiyah Purwokerto. The Honorable Dean of Pharmacy Faculty, Universitas Muhammadiyah Purwokerto, Mr. Didik Setiawan, PhD. The Honorable Dean of Pharmacy Faculty, University of Szeged, Professor Isvan Zuko. The Honorable Professor Piroska Shaborifes is our guest lecturer today. The Honorable all lecturers of the Pharmacy Faculty UMP and the Honorable all participants of this lecture. I'm Arini Sharifah. I am glad to be the moderator for this guest lecture entitled Pharmaceutical Preformulation of Drug Substance, Crystallization, Amorphization, and Complexation. Ladies and gentlemen, please allow me first to read agenda for today. The first schedule is the opening session, and the second schedule is lecture session. We are now in the opening session. Without further ado, I would like to invite Professor Isvan Zuko to give the opening speech from University of Szeged, from Faculty Pharmacy of University of Szeged. To Professor Isvan Zuko, the time and screen are yours.
Thank you so much. So, dear colleagues, ladies and gentlemen, it is my privilege, my pleasure to welcome all of you from Szeged, Hungary, on behalf of the staff of our faculty. Our two countries and our two faculties are far from each other, but I am deeply convinced that in spite of the geographical distance, our interests and our motivations are very similar or the same. And that's why I believe that we can work together in terms of education and research activities. That's why I hope that this lecture delivered by Professor Piroska Révész is not simply a lecture which is a comprehensive summary about one of the research topic going on at our Department of Pharmaceutical Technology, but maybe a first step in our cooperation. So, colleagues, hopefully we will continue with versatile topics and hopefully some student and academic mobilities will be also started between our two faculties. I'm really grateful for the organization of this online event. So many thanks for the staff of your university. I am grateful to Professor Didik, and I am also grateful to Professor Piroska Révész for this lecture. Dear colleagues, thank you so much.
Thank you, Professor Isvan Suko, for your speech. And then the next is the second opening speech from Dean of Faculty Pharmacy, Universitas Muhammadiyah Purwokerto. I would like to invite Mr. Didik Setiawan, PhD, to give the second opening speech. Mr. Didik Setiawan, the time and screen are yours.
Thank you very much, Mrs. Arini. Again, I'd like to say thank you. Willingness to and also warm welcome to from for our hope event we can have another collaboration activities like Professor Subko mentioned previously, that we can do a research collaboration or more student or staff mobility between two institutions, and hopefully we can make it works in the next few months. And special thanks to Prof. Piroska and also the committee from the Pharmaceutical Technology Department who are successfully create this event, and hopefully a lot of participants who can they can learn a lot of things from Prof. Piroska, as we know that her expertise is outstanding. So for the student, please enjoy the lecture in the next few hours. Hopefully you can learn a lot and can give you it can give you an inspiration in terms of research. Maybe you can choose one of the you can choose it as your topics in your final thesis. Maybe that is from me, not that much, so we can learn a lot from Professor Piroska. Thank you very much, and good luck everybody, and enjoy the lecture.
Thank you, Mr. Didik, for your speech. We will move to the next agenda. The following agenda is case lecture. Ladies and gentlemen, our speaker today is Professor Emerita Piroska Saborives. Wait a minute, I will have problem. Okay. She is a lecturer in the Faculty Pharmacy at the University Szeged, Hungary. She received her doctoral degree in the Medical University of Szeged and became a specialist pharmacist, especially in the pharmaceutical technology field in 1979. She has been head of the Department of Pharmaceutical Technology of ST for 2005 until 2016. and a professor of Department of Pharmaceutical Technology and Regulatory from 2016 until 2018. And then she received many recognition during her professional career. Among them below are some of the most recent: the Claesberg-Kuno Award in recognition of achievements in the field of science and education, the honor of Claesberg-Kuno Foundation, University of Szeged in 2017, and Major Edmund Lopageres Prize from President of the Republic Hungary in 2015. Her research field is the modification of the physicochemical properties of active agent, crystallization, amorphization, nanonization, study of the drug-like behavior of pharmacons, design of formulation for clinical studies, target therapy, and development of micro and nano carrier system, particularly for nasal and pulmonary delivery. From this research, she has published 387 papers, 2,250 citation index, more than 200 impact factors, 2 patents, 4 inventor, 10 books, chapters, 3 university handouts, and 1 book in editing. Wow, this is the great achievement. Okay, ladies and gentlemen, we must know preformulation before formulating drug or active pharmaceutical ingredient into a dosage form. Every drug or API has chemical and physical properties, but today we will talk about physical properties like crystallization, amorphization, and polymorphism. And ladies and gentlemen, one of the exciting things from Piroska's journal about the importance of polymorphism. It is increasing because of its value in the pharmaceutical industry for various reasons. What is crystal, amorph, and polymorphism? How is it urgently needed at the time preformulation studies? Without prolonging time, we will listen to the lecture and find the answer to this question. To Prof. Piroska, time and screen are yours.
Okay, thank you very much. Do you hear me?
Yes, I hear you, Prof.
Can I begin my presentation?
Yes.
Yes, sure,
Okay.
Professor.
Okay, thank you very much. Good afternoon, dear colleagues and students.
Good afternoon, Prof.
Okay?
Yes, I hear you clearly, and your presentation show in my Zoom meet.
Okay. Welcome, everyone. First of all, I would like to say thank you very much for the invitation to hold the presentation with the title Pharmaceutical Preformulation of Drug Substances, Crystallization, Amorphization, and Complexation. Just a minute. I cannot step. Okay. In my presentation, I would like to say about our country, university, our faculty, and I would like to show you the background of these topics, and I would like to talk about our research work. As you know, I think as you know that Hungary is located in Central Europe, and the number of the population is about 10 million, and Budapest is our capital. Szeged is located in southern Hungary, and it's a very nice town. It is the city of sunshine and students. Our university has 22,000 students, and we have famous Professor Albert Szent-Györgyi, who had the Nobel Prize in Physiology and Medicine, and he isolated the vitamin C, and he discovered the components and reaction of the citric acid cycle. Our university has 12 faculties. Our faculty has about 500 students. We have a Hungarian and English language program, and the doctoral school has 50, 60 persons or students, and it works very well. You can see the program of this doctoral school, and later you can choose from this program, as mentioned our dean. My institute is Pharmaceutical Technology and Regulatory Affairs. We have different research topics. We have excellent instruments park, up-to-date different testers. We have industrial partnership and cooperation with domestic and foreign partners. We have 15 PhD students, and we have a lot results which is published in excellent journals. And my first topic, research topic for today is the preformulation development, and I mentioned the crystallization, amorphization, and complexation. But our department or institute has another programs, for example, the solid pharmaceutical development, dermal, transdermal application, nasal, pulmonal administration route. It is related to the particle engineering. We deal with the nanomedicine and the patient adherence in chronic disease, and we deal the regulatory science models, which is related to the quality by design methodology. And the first part of my presentation is the preformulation of drug substances. What does it mean, the preformulation? Preformulation of active pharmaceutical ingredients. We know that we have different drug APIs, for example, with chemically synthesized agents, semi-synthetic substances, and we have the active agents produced by biotechnology with the medium molecular weight as insulin, or with a high molecular weight as monoclonal antibody. If you see this figure, it is the analysis of APIs approved by the FDA. You can see here that we have not too much active agents, new active agents per year. And from these, the chemical entities is more than the biologic APIs. And I thought that my presentation has, first of all, the chemical agents. I will talk about the preformulation of chemical agents, and the biologic agent is also a very big topic, and it is maybe the topic on another presentation. Here you can follow the distribution of the new active substances. You can see here that first of all, the pyrazoles and fluorinated as chemical active agents are in more as opposite to another active agents. And it's very interesting that according to FDA and EMA, the therapeutic field is same: oncology, hematology, infection, and we think that later in the future we have more and more chemical entities, and of course plus biological agents also. The preformulation of drug substances means the change of physical chemical properties of these APIs according to manufacturing aspect and biopharmaceutical aspect. You can see here that we can change the particle size or the particle size distribution of the agent, or the crystalline and amorphous ratio of API, and we can change the technological parameters as bulk density, flow properties, compression properties, and the stability also very important, and we can change these parameters to have a better condition for manufacturing of API. And the biopharmaceutical aspect also very important in case of preformulation study: solubility, dissolution rate, lipophilicity, permeability, log-D, and the polymorphism also. The final goal is to research and develop a new technology, innovative new technologies, and design the new dosage form or the new testing method. If we talk about the preformulation of active agents, we know that very important the preformulation of the pharmacon candidate. But I mentioned that we have not too much pharmacon candidate. Therefore, we preformulate the already known active agents. We can prepare the value-added products, or we can design the unmet medical need products as drug device products and inhalation preparations, and we can choose the administration route. In case the value-added products, I would like to talk later about the modification of dissolution rate or solubility, and change the bioavailability of the active agent. Before we design the experimental work, in the case of a preformulation study, we should have information about the guidelines for active ingredient production, because the drug master file has such information, which is maybe very important during the research work or the development. It is same in case of final dosage form. We have a common technical document which has also the very important information for manufacturer, for control of critical steps and the description of manufacturing, for validation. And finally, we should have the new guidelines for industrial production, for active agent and for dosage form products, for example, production, for example, the new dosage form, for example. It is a critical process parameter. We must optimize the critical quality attribute. We should have a design pace parameter, hold time. We use the real-time release testing, and we use the PAT and the QBD. There are new relations in this topic, but I think this is very important to the research work and later the development work. It was introduction, and now I would like to talk about the crystallization as a basic industrial process, which is very important because of purification of active agent, to give a good quality crystals, change the particle size distribution. We can prepare desired polymorphic modification, and we can influence with the new crystalline form the biological effect. I think you know the thermodynamic aspect of the crystallization. The driving force is very important. Is a difference between the two phases as current supersaturated concentration as phase and equilibrium solubility concentration. If our system has a lot energy, the difference between two phases in concentration, we can drive this process that we have different particle size or our final product. The crystals have a different particle size, and the other parameter, we can change the other parameters. You can see here the solubility curve. It is well known. I think here the solubility curve and super solubility curve, and between two curves, we can drive the crystallization in metastable region. Here you can see the reactor for crystallization and the very conventional crystallization processes from solution as cooling crystallization, salting out crystallization, and reactive crystallization by precipitation. We optimize the process parameters, and we can have the final product. And now I would like to show you two samples. It is as the practical relevance. How can influence the steering rate, the particle size of the final product? You can see the paracetamol crystals. Cooling crystallization was used. The solvent was 2-propanol. And we can follow here that the steering rate if we increase the stirring rate, the particle size of paracetamol will be decreased. In case of change the co-solvent, it is resulted the plate crystals. In case of water as co-solvent, we have a bigger crystals as in the case of ethyl acetate. But we know that the paracetamol is used in tablet form first of all, and the single dose or the pro-dose amount is 500 milligram. It is too much to tablet making by direct pressing. If we use these crystals, we must use the wet granulation before the tablet making, and it is not so practical, and we have some problems during the tablet making. Therefore, we look for the new and newer crystallization processes which resulted the product with better flowability and compressibility. And another example here to this part is stearic acid as lubricant is used for tablet making. And you can see here that we have 3 different batches which has different parameters as a specific surface and the particle size distribution. If you see the batch 2, in case of batch 2, we have bigger particles, the surface of particle very smooth, and the specific surface is less than in case of batch 1. It means that the batch 1 has a bigger specific surface and the particle size distribution is another than that of batch 2. It means that the mixing time, if we use these components, may be very different. In case of batch 1, the mixing time is 1 minute, and for batch 2, the mixing time is 10 minutes. It's a very, very different. And if we use the if we use first or in the common technical document is a mixing time as 1 minute, and later we have or we use batch 2, the mixing time will change and it will be a problem. And the conclusion is very simple. Always we must use the same quality product. It may be a problem during the tablet making. So after this introduction of crystallization, I would like to show you the special crystallization processes from our research work. It is a spherical crystallization by spherical agglomeration and quasi-emulsion solvent diffusion method. And we can decrease the particle size by starting out crystallization with impinging jet technique. It is a self-made technique. Why is important the crystallization, the spherical crystallization? I mentioned the paracetamol that if we have the paracetamol crystals with unfavorable habit parameters, we must use wet granulation during the tablet making. If we use spherical crystals, we can use a direct pressing, and we can change also the unfavorable habit. We can increase the particle size. It results in good flow properties, and we can use a high dose of active agent during the tablet pressing. The spherical crystallization was, the process was published by Kawashima in 1982 in Science, and it was a very, very important first publication in spherical crystallization, about the spherical crystallization. Kawashima defined the spherical crystallization. You can see that an agglomeration process that organizes the material into the compact spherical form during crystallization. They use the QESD method, which means that they work the 3 different solvents, and model material was salicylic acid. The good solvent for salicylic acid was ethanol, the non-solvent water, and the chloroform was the wetting agent. During the process, they have emulsion drops, and the good solvent left the emulsion drops and formed spherical crystals. Our research group in 2000 start with the spherical crystallization. First, we crystallize the acetyl salicylic acid by emulsion, quasi-emulsion solvent diffusion method. You can see here the final product, which shows a spherical form. The good solvent was ethanol, the poor solvent water, and the bridging liquid carbon tetrachloride. It is very important that the bridging liquid in our work, and in case of Kawashima work, belongs to the class 1 as danger class according to ICH. It was a very big problem. Therefore, we looked for another possibility to have or to produce the spherical crystals without danger or the very problematic solution, instead of very problematic solution. In case of magnesium aspartate, we used 2 solvents. The good solvent was water, and the poor solvent the methanol. We used the starting out crystallization process, and the final product was a spherical agglomerate. And the process was very simple because during the starting out process we had aggregates or the precipitated crystals, and here the bridging liquid was the water, the liberated water. It means that the water molecules stick on the crystal surface, and during the drying process it was solidified, and the solid bridge create or form the spherical agglomerates. You can see here that it's very simple process, and the parameters and the final product and the parameters was very good. First of all, the geometrical parameters and the roundness value was also very good. The optimal roundness value is 1. In case of developed product, it was this parameter near to 1, and the specific surface was also smaller than the control product. And if you see the next parameters, it's powder rheological parameters, you can see here that the CAR index, which is calculated from pore density and tapped density, shows excellent good flowability for developed magnesium aspartate. It's 5.32, and for acetylsalicylic acid particles or CARS index, it was also about 6. It means that this raw material produced or crystallized by spherical crystallization are suitable for direct pressing. We have this DC name in the name. For example, DC magnesium aspartate. It means that direct compressible magnesium aspartate or acetylsalicylic acid. And later we looked for another possibility to have spherical crystals, and now we know and we can use the other crystallization processes near the quasi-immersion method and spherical agglomeration method. For example, the cooling crystallization with alternating temperature profile. You can see here the crystal agglomerates, and we use only 1 or 2 solvent, and these 2 composition, 2 solvent composition is suitable for preparation of spherical crystals. And another new possibility for crystallization, it's impinging jet crystallization. With spherical crystallization, we can increase the particle size, and with this process, the impinging jet, we decrease the particle size. It's very important because the conventional crystallization or crystallizations are not suitable for particle size decrease into micrometer range or nanometer range. Therefore, this possibility is a very new crystallization process. Here we use, you can see here this tool as an impinging jet, which is in the reactor, and it has such tubes which is suitable to meet the saturated solution and the antisolvent. Here in this place, at this place or at this point, we have the nucleation and the crystals very small. This system hasn't enough energy to increase the particles because we have a lot nucleus in the system. The model drug was glycine in this project, and we used different ratio of solvent, and the crystallized product has a smaller particles and the other conventional methods. You can see here the other conventional methods. First of all, the cooling, and after that the reverse anti-solvent method and anti-solvent. And you can see here that the impinging jet resulted in small particles between 8 and 35 micrometer, and the particle size distribution was very good. We can continue this technology, and we can decrease technology, but we can reach, it's my opinion, we can reach the nanosize range, because the limit of nanosize range is 1 micrometer. It means 1000 nanometer, and this nanometer range, for example, between 100 and 1000, is a very critical particle size, and we cannot use such crystallization which can result this particle size, or we can use the technique, but with polymer or other auxiliary material, which may be impurity in case of final product. It is other topic, but in case of conventional crystallization, the industry, the pharmaceutical industry, must produce raw product without any impurity. So, and here I would like to show you that we have another crystallization topic in the industry, but this topics, crystallization processes, is used in cooperation. It is a supercritical fluid technology, or the application of alternative energy in crystallization as electrospray and cold plasma crystallization, as well as cooling crystallization in electrostatic fields. And we use the sonocrystallization also. And if polymorphism, or if crystallization, then polymorphism. If we use a crystallization process, we should calculate with the polymorphism. Here I would like to show you the polymorph screening, because it is it belongs to the preformulation, and it is very important, first of all, in case of new active agents, but in case of old agents is also very important, because we can find the newer polymorph modification, which may be interesting for pharmaceutical application. We have the possibility to reach the Cambridge structural database, and we can check in this database the different forms, and our instruments park is suitable for polymorph screening. Here I would like to show you the protocol, only the steps. If we have a pharmacon candidate, first we should generate the different polymorph modification using different solvents and transformation. We use, for example, the heating transformation. And if we ready with the crystallization, we have very small crystals. The next step is identification, and then the relative stability examination. You can see here the different methods which are used to identification the polymorphic modification. In case of pharmacon candidate, it was an industrial project. We found 8 polymorphic modifications with amorphous form, and we use the transformation. Altogether, we found that we have 3 polymorph modification which were stable polymorphic modification from 2, from 3, and form 4B. The SEM pictures show the different habit, and if we see the dissolution rate, which is important later, you can see here that the 3 stable polymorphic modification here down showed the very slow dissolution rate. The structure was stable, and the stable structure resulted slow dissolution rate. This pharmacon remained as a candidate, and we cannot follow this process during in time. And here I think it may be interesting, this history about the patentability of the new crystalline form, which belongs to the NutraSuite, and the new polymorph modification. The Aimoto Corporation purchased the license of this material, the sweetener agent, from Monsanto company, and during the crystallization they found other crystalline form. They used only the slower crystallization process, and without mixing they found the new crystal modification, which was favorable, and it was the question, it is a new polymorph modification or not. The Aimoto Corporation won the lawsuit. And I think here is maybe the comment, it is the wars for new, not yet protected forms. And at the end of crystallization process, I would like to show you what is the new trend in this field. We use PAT technology, the process analytical technology. We can detect the end of reaction, or we can use the feedback control crystallization. If we use a different tester as Raman, as FTIR, and FB-RM control, which shows the particle size distribution. Here, I would like to show you the last tester as the focus beam reflectance measurement, which is suitable to follow the particle size change in the reactor in the crystal suspension by laser beam. And this tester gives parameter for chord length, and it is provides a particle number, fiber length distribution, and we can have different curves. For example, the first figure shows two curves in case of seeding process during, or the add antisolvents during the crystallization, or in case of time, we can follow the particle size distribution. It means that during the preformulation study, we optimize these parameters, and during the development of the final crystallization process, we can compare the data and we can follow what's happened in the reactor sphere. Or the Raman spectrum, we use this possibility if the agent has a different polymorphic modification. And what is the new challenge in this topic? I think the new challenge is the continuous pharmaceutical technology, and PAT and QBD, but I know that this process and the requirements are not simple, and the colleagues in the pharmaceutical industry work a lot to have or to give or to produce excellent good products by crystallization. And here the next topic, but the time is short. I would like to talk about the amorphization, but I think it's very well known topic. First of all, I would like to show you the examples which is connected the amorphization. Why is important the amorphization? As you know that we prevent the API from the interconversion. We use this possibility in case of water, poorly solubilities, drug, or in case of if the process is difficult, or we can get the new patent. Sorry. What is the difference between the crystalline and amorphous form? It is well known knowledge, and we have good glass former materials and poor glass former materials. In case of sugar alcohols, the magnet is a poor glass former material and the sorbate is a good glass former. We can calculate the TG as glass transition and TM ratio, and if in Kelvin, and if this value is 0.70 or less than 0.70, our API shows poor glass former property, and if this value is 0.70, we can work the API which has a good glass forming property. Here, show you the technologies which are used during the amorphization as solvent, milling, and hot melt technology. I would like to show you the example to connect these technologies. The stabilization of the amorphous API, we know that without stabilization, we cannot cannot prepare a stable product. My opinion is that the very good stabilization possibility is a solidification of amorphous API, but in case of solidification, we should use also crystallization inhibitors too. Crystallization inhibitors, crystalline and semi-crystalline and amorphous auxiliary materials. First of all, the cellulose derivatives has a very good property to fix the amorphous API and has an interaction with API, the amorphous API. And example, the first example is preventing of the interconversion of polymorphs through amorphization. The problem is transformation of different modification, and here it's very, very important. The requirement is totally amorphization of API. It's very, very important, and we have a critical value for crystalline material is the amount of crystalline fraction is below 1%, and we can measure the crystallization fraction amount. We use DSC and XRPD method. And example, the clopidogrel hydrogen sulfate as good glass former material, and it has 6 different polymorphic forms, and we use in the pharmaceutical preparation the form 1 and form 2, but because of interconversion, we have the amorphous form, which I mentioned the totally amorphous. We prepare the totally amorphous form. And the steps, I see I have a question, but after this part, I will check it. We look for the solvent, which result the amorphous form. It was ethanol, and we made a preliminary stability test, and we found that our amorphous clopidogrel hydrogen sulfate will recrystallize during the time, and it is unstable. You can see here the storage day and enthalpy value. And the next step, selection of crystallization inhibitor. Here the DSC curve and the melting point of activation, and the B form is an amorphous form of API. We found that it is a B is an amorphous of a reference sample, and we found that the aerosol silicium dioxide was the very good material which stabilized the amorphous API. And then the determinant of the amount of the crystallization inhibitor, it was ratio clopidogrel silicium dioxide 7:3. And you can see here the XRPD fingerprints for crystalline API, for amorphous API, and amorphous API with aerosol. With aerosol, which shows also amorphous product. And the tablet was produced with this amorphous API, and the FTIR investigations show that we have an interaction between the hydrogen bond, interaction between the silanol groups of aerosol and the hydroxy groups of NCC, and the tablet was very stable, and the recrystallization was not detectable during the storage. And another possibility for amorphization is a milling process. In case of milling process, the first aim is a particle size, requirement of required particle size, and we don't use or reach the amorphous form, but with the decreased particle size, for example, nonionization, we must calculate the amorphization of the API. The crystal structure breakdown, and the product has a crystalline and amorphous forms, and the crystalline part works as seed crystal, and the amorphous structure can generate supersaturation, and it can result in the crystal precipitation. Here you can see how can we calculate the crystallinity degree by XRPD, TST, NIR, and very important information that the industrial gold standard for investigation of crystal structure is X-ray powder diffraction. Here the slide shows the milling process, and we can see here in time the particle structure changing, and it is important that in case of mirroring process, the particles, the crystallinity degree increase according to exponential model. So, and another example here to decrease the particle size and with increased amorphization of API is a wet milling technology. Here we have N-side material as meloxicam, which belongs to the poor glass former class. And we use the polyvinyl alcohol as protective polymer, and the critical parameter was the particle size range between 100 and 500 nanometer. If we reach this particle size range, we can control, or during the process, we can control the crystallinity degree, and after that, we can use or select the stabilization process, as here in fluidization, lyophilization for solidification of the product. And we can use the other auxiliary material for stabilization. You can see here the process. We use the wet milling. I mentioned it was a planetary ball mill combined with power milling. The optimized process parameters are here, and the predispersion had meloxicam and polyvinyl alcohol as the protective agent. The nanosuspension with MEL had such parameter which was required, 130 nanometer, and this nanosuspension was solidified. In case of fluidization, the carrier was microcrystalline cellulose, and in case of lyophilization, trehalose. Here you can see here the particle size, the average particle size for nanosuspension. It was so liquid, and here the holding time was only 2 weeks, but we used the suspension prompt for solidification, and we didn't wait the storage for the storage, and the fluid MEL and the lyomel showed the same particle size, average particle size, but in case of two product, the carrier had on the surface the meloxicam, and we used here the SEM image technique. And you should know the lyophilization process is almost long or longer process as fluidization. And the next slide shows very good that the crystallinity degree of nanomel and the fluid mel was same. But in case of lyophilized meloxicam had a particle size increase because of longer process time. And the storage showed a very good result for fluid mel. And in case of lyomel, didn't change the crystallinity degree. And the cell viability test was excellent, good. The components doesn't show any toxicity. We used here the CACO2 model, and here the in vitro, in vivo results. We had correlation, IV-IV correlation between two methods, and which is very important and interesting here, that the nanomel nanosuspension showed not too good result here, and the other two methods, the fluid mel and lyomel, had a better AUC value, the bioavailability, because of the surface had the particles, and the distribution of the API was very good, and we think that the nanoparticles from nanosuspension can be aggregated in the stomach. Yes, and here the MEL technology in the last in amorphization. We use here in the institute, first of all, the pastille hot melt technology. First of all, we developed the Lozenge pastille. The carrier material was a eutectic mixture with xylitol and mannitol, and the pH was polyethylene glycol was a softener agent. These pastilles have here the paracetamol, and in case of therapy or the in vitro study, this product showed a very good in vitro dissolution. And hot melt technology as extrusion is very important in this topic, and the pharmaceutical industry uses this technology, and with different polymers, and they have a very good results with this technology. And the solidification of melt in drops from pharmaceutical industry, it is the practical relevance of the melt technology. And before the complexation, okay, I'm in time. I would like to give an answer. Just a minute. Okay. Okay, I see. Okay. So I continue my presentation. And the last topic here, the complexation. I think the three different topics were too much together. And I thought that the complexation will be a very short part of this presentation. We know that there are different possibilities for complexation, but allow me to talk about the Hungarian-related complexing material. The cyclodextrin is a very famous agent, carrier agent in the pharmaceutical industry, and we can use this agent in preformulation study, and we have or had a very famous colleagues, József Szejtli, who was a chemical engineer and who started the cyclodextrin production or research in Hungary. You can see here that he founded the company in Budapest, and nowadays this company work as Cyclolab, Cyclodextrin Research and Development Laboratory. We have a very good cooperation with this laboratory, and we have the new and newer cyclodextrin type to use research work and for development of the different dosage form. Oh. I cannot. Okay. A few sentences about the cyclodextrin, but first of all, I would like to show you that is important because of we can form with cyclodextrin inclusion complex, we can increase the solubility. It's a very important because the solubility is sometimes not only very important, but we cannot, we could not use the other carrier to increase the solubility, and we can increase the stability of the API. And we know that the cyclodextrin have an interaction of lipids, membrane lipids is very important, and the new information is that the cyclodextrin is used in Niemann-Pick disease type C, and the hydroxypropyl beta cyclodextrin works as active agent in this therapy. Here I would like to show you some data or research work data about the solubility enhancement property of API in case of loratadine. You can see here that the dimethyl beta cyclodextrin was very good as solubility enhancer, opposite to loratadine, which showed the 1 unit as solubility enhancer, and here with the dimethyl beta cyclodextrin, it showed 3,000 enhanced fold as solubility enhancer. And we checked the blood concentration in animal test, and we used the edema test, and we found that the dimethyl beta cyclodextrin increased the blood concentration. If we change the ratio, we have more and more blood concentration, and in case of edema, we found that decreased the edema volume, and the beta cyclodextrin, dimethyl beta cyclodextrin didn't influence the extent of edema relative to the control group. It's very, very important. And the next work was the gemfibrozil dimethyl beta cyclodextrin inclusion complex preparation or production by co-grinding method. This work is interesting because we use a very different test to identify the inclusion complex, XRPD and DSC, but we had and we published first the FTIR curves, which was suitable for measure as analytical method for measure the discriminate of amorphous and inclusion complex present in this system for gemcitabine. And the last topic in case of complexation is a terbinafine hydrochloride inclusion complex. It was prepared by co-grinding. You can see here the phase solubility curves or diagrams, which shows that the dimethyl beta cyclodextrin shows higher affinity to the API, opposite to the other cyclodextrin type, and the XRPD diagrams show the amorphization at 30 minutes for dimethyl beta dioxide, and hydroxypropyl beta cyclodextrin shows also such inclusion complex which shows amorphous character. And you can see here the products, the co-grind products, and the particle size, average particle size, and the in vitro dissolution, which shows better dissolution properties for both cyclodextrins. I think this investigations, this production of cyclodextrin and investigations shows or show the possibility to use this carrier material in case of other API which belong or belongs to BC2 class, they have a poorly water solubility. And finally, I would like to show you this picture, and I would like to talk about that we use our research results and our knowledge, first of all, to preparation, design, and development of different dosage form for alternative administration routes as nasal and pulmonary administration. Our model drug is NSAIDs drugs. You can see here a few NSAIDs drugs, and we target the brain, nose to brain, first of all by nasal administration route, but it's important the systemic therapy, the pain relief with nose to blood. And in case of pulmonary therapy, first of all, we use these NSAIDs for cystic fibrosis and non-small cell lung cancer. For local therapy, first of all, DPI form, and we have some results and a lot, I think, a lot publications. You can see these publications, and later I think it may be a good cooperative possibility, these topics and basic topics as crystallization, amorphization, and complexation. And the conclusion, the topics offer a number of additional research opportunities, and I think that we can have innovation in case of R&D of technological processes, and also in case of products. If we have a good results, we can prepare a value-added preparations. We are open to cooperation, and finally, I would like to thanks for the attention, and I wait for your question or comments. Thank you very much.
Thank you very much, Prof. Piroska, for giving us such informative and interesting material. Ladies and gentlemen, now come to the question and answer session. I want to divide three sessions and have three sessions and have three questions from the participant each session. You can give question by typing your question in box chat, or you can ask directly by raise hand. Okay, Prof, we have one question. Okay, I will translate. This is a question, but he asks with direct question, with direct message, or maybe we move to the next question direct because she want to ask question directly, Prof. You. Okay. This is my. Okay. She is my student, Audita.
Okay, hello, Mrs. Arni. Thank you for opportunity for us. Hello, Professor Piroska. Excuse me, I am Audita. Yeah. I am Audita. As undergraduate pharmacy student of Universitas Muhammadiyah Purwokerto. Excuse me, Prof. I am interested in research and development of other crystallization processes. Especially, I want to know why cut plasma crystallization can be used for application of alternative energies in the crystallization. I saw this is in description slide 34. Thank you, Professor.
And the question is belongs to crystallization. Yes?
Yes.
Crystallization crystallization and the crystallization method. I don't understand well the question. The energy or the.
My question is why cold plasma crystallization can be used for alternative energies?
I see. I see. Okay. The alternative route I mentioned, maybe the nasal and pulmonal, and if we have API not or the unfavorable particle size or habit, we can change the habit by crystallization. For example, we can decrease we can decrease the particle size by crystallization, and if we use the crystallization, we have pure crystals, and in case of pure crystals, we can use in suspension, for example, or we can decrease the particle size into the nanoparticle size range, and we use crystallization process. It's for example, a bottom-up method as nanocrystallization method, but in case of bottom-up method, we must use the auxiliary agent. So, do you understand my answer? So, we change the particle size with the conventional crystallization method, and we use the preparation of nasal dosage form, or we can use nanonization, and we can use also through the nasal application, but the small particles or are suitable for target the brain, nose to brain, for example.
Okay, Prof. Maybe Miss Audita has a problem connection. So we move to the next question, Prof. Okay, the next question from Fatia. You can ask directly to Professor Piroska.
Yeah, okay. Hello, Mr. Rita. I'm Fatia Nurstautami with student number 111 from pharmacy. I want to ask about how to we make sure that the drug is complexed in the cyclodextrin. You got my question?
It's a complexation.
The complexion, how to make sure that the drug is already complex in the cyclodextrin. Yeah, how to make sure it.
The complex formation we measure by XRPD, first of all, and the FTIR. If we have an inclusion complex, the API is in the a cavity of cyclodextrin in molecular form, but the one part of API may be amorphous form near the inclusion complex. So we can measure the crystallinity. I mentioned that we have an article which shows how can we measure together these forms by FTIR, and we can see that in the publication that we have a characteristic peak for cyclodextrin and for API, and the API peak measured by FTIR will decrease, and CD complex, inclusion complex, will increase, and the API will decrease. Altogether, we have transformed curve, which will shows semi-quantitative, how many is the inclusion complex, how many is the amorphized API, which isn't in the cavity of the cyclodextrin. I'm very sorry, but I wanted to bring this figure because it's very important, and it was the first present by colleagues, but you can find on the slide if you have my presentation.
Okay. Are you get it the explanation, Fatia?
Yes, Miss. Thank you so much.
Okay. Yeah. Okay, and then this is the relatable with the previous question, Prof. This is from Bagaskara. He asked with a private message by me. He now doing about cyclodextrin formulation, Prof. He did with ibuprofen, and he asked about. I'm sorry. He asked about what is the effect time and then the effect of temperature when he what is it? When he makes cyclodextrin. Is it true if heating temperature heating and then the time can be effect for cyclodextrin? What is it? Is cyclodextrin form? Prof? Are you
Yes,
get it? Yes. Are you get it this question? Yes.
Yes, don't understand because the contact not so good.
Yes.
So, but I think that the question was cyclodextrin and the preparation of inclusion complex and how can influence the preparation of formation, the complex, the temperature.
Yes, that's right.
Yes. I think it's important parameter, the temperature, but in case of preparation, we use in the institute, in our institute, the kneading product, and milling product. The kneading product means that as wet milling process, and dry milling process means that we have a cyclodextrin and API, and we use the mortar and pestle technique. It was a laboratory scale, and we found with this technique the product, and we didn't measure the temperature during the process, but I think it's very important parameter. Yes, we have the cyclodextrin sample in small amount in general, and it is suitable only for production of production of small amount of product. We use in laboratory scale. So, but my answer is the temperature is very important.
Okay, and then how much the temperature, Prof, to make cyclodextrin?
How much?
Yes, sorry. Maybe how temperature to make cyclodextrin, to make perfect cyclodextrin? Maybe about 30 Celsius, maybe.
Sorry, my question is my answer is I don't have any data about it. Yes.
Okay, no problem, Prof. Okay, we move okay, no problem, Prof. We move to the next question. Maybe we have a money question, Prof. The first, maybe ladies first, yeah. Mas Oi after Mbak Diandra. Next, Mbak Diandra. Maybe you have bad connection. Sorry, Deandra, maybe you have bad connection because I don't hear you clearly.
The connection isn't so good.
Yes.
Oh, sorry, I have a low connection. Maybe I repeat my question. My question is, what are some of the challenges we face it when we use natural help and regular API use it in formulation, and which process is recommended to upgrade renovation in because our country is developed of technology.
Sorry, Deandra, maybe I will postpone your question because your connection too bad. So we move the next question. Yes, Mas Oi, you can ask directly with Professor Piroska.
I don't understand the question because of the connection isn't so good.
Yes, me too, Prof.
Yeah. But I suggest you that you can see here my email address. You can ask me, okay? Write please email, and I will give answer for your questions. Okay.
Okay, you can ask with Professor Piroska with sending question with this email.
Yes.
Okay.
Yes, because the contact is very wrong. Yes,
okay.
I don't understand you, but
okay. Yes, me too, sir. Me too,
yes,
Miss.
Okay.
Okay. Next, Mas Ai.
Excuse me, madam. Let me introduce myself. My name is Oi from Pharmacy 7 semester, bachelor degree. I want to ask about preventing of the interconversion of polymorph through amorphization. How to know concentration of crystallization is below 1%. What is the side effect if the crystallization is above 1%? Thank you.
During the amorphization, the crystalline phase may be, I mentioned, 1%, or less 1%. If this value is higher, we have a recrystallization. This a few crystals can work as seed crystals. But how can we measure or test this amount of crystalline phase? It's a very good question. The XRPD doesn't suitable for measure of small crystallinity degree. If this value is less than 5%, the XRPD didn't see the crystalline structure, and we cannot use the XRPD for characterization of crystallinity degree. In case of, for example, NMR is a suitable for characterization of API if the crystallinity degree less than 1%, and the side effect is it's sure that the recrystallization if this value is more than 1%. we had industrial project, and the pharmaceutical industry measured the crystallinity degree during the storage by XRPD, and I mentioned that the gold standard is XRPD for measure of crystallinity degree.
Okay, are you get it the answer, Mas Oi? Ya, jadi kalau kalau mau tahu berapa konsentrasinya bisa pakai NMR. Kurang lebihnya seperti itu. Okay, baik, we move to the next question. Okay, this question in chat box, Prof. This is from Muhammad Novianto. What is the main differences between the formulation of the common drug or maybe chemical drug with the formulation of the natural herb drug? Because do we have to consider about the crystallization and amorphization and complexation, especially for natural drug, Prof?
It's a very good question, but I think it may be the crystallization of natural drug, but it's sure if we would like to have a stable form, the crystal structure is important, and the crystallization also important in case of natural drugs. But I think that the natural drugs can have the other components, and this mixture or the other components can change the structure of the natural drug, and it may be amorphous, can show amorphous structure. I think that it may be use complexation, and I remember we used the complexation in case of natural drug because it was unstable, and the cyclodextrin stabilized the natural drug. So first, the crystalline form, crystallization, and the second one, I think the use of cyclodextrin and the amorphization. It may be it may be suitable, but we should calculate with unstable structure or the final product. But it's my answer.
Are you getting the question, Mas Novianto? Okay, we move to the next session. The next session, like before, is divided into three questions. Yeah, for the first question, yeah, Mbak Rosi.
Thank you, Mrs. Harini. Hello, madam. My name is Rosiana from Master Degree of Pharmacy. I want to ask about, yes, I want to ask about preventing of the interconversion of polymorphs through amorphization. I want to ask about the point about the stabilization with crystallization inhibitor. How stabilization with crystallization inhibitor can prevent of the interconversion of polymorph through amorphization? Thank you.
First of all, we use polymer as a crystallization inhibitor. And we know that the polymer can have such functional group which has a contact with the API. Between the contact between the API and the polymer, may be such strong, which protect our amorphized product, and during the storage we cannot have any changing. But I think the conversion of the amorphous structure not so stable with the only polymer. We use the solidification of the product. It's very important, and it's very important the other auxiliary material which has a very big specific surface, and in this case during the interconversion of API polymorphic modification through amorphization, we have amorphous API, we have a polymer which or other material which has a direct contact with the amorphous API, and we must use the carrier as microcrystalline cellulose, for example. And on the surface of the microcrystalline cellulose will fix the amorphous API with the polymer. It means double fixation or stabilize of the amorphous form. This product, according our results, showed a very good stability, but the storage time it is shorter than that of the crystalline material. So about 1 year, the final product we can store and use or is on the market 1 year. If we use a polymorphic modification of crystalline form, it may be longer, or it's sure it may we can use longer time. Okay.
Thank you, Professor.
Okay. Okay, and then the next, sorry, ladies and gentlemen, I will select it on the question because we have several questions, and then I will select it question. So if your question don't answer, I will what is it? I will make summary question, and then I will email to Prof. Piroska. Would you like to answer the question, Prof? If there are, because we have limited time.
Igen. Még most beszél, de nem értem. Igen. Ja, jó, okay. I wait for your email, and I will answer your question.
Okay, so the next question, this is from Elok Nabila. I would like about crystalline polymorphism in a compound, because because before in polymorphism, maybe I will example with glycerin, Prof. Before you said about glycerin have 8 polymorphism forms. So what is different? What is the effect? What is the 8 polymorphism have similar or not similar bioavailability, Prof?
Yes, the glycine is an amino acid, and it has 2 polymorph modifications, and the polymorph modifications shows the different habit. but we use the one polymorph modifications, but I don't know now what is the correct answer in this case, because I don't know very well this topic. Yes, it was very interesting because of the impinging jet, and there we used or we had one polymorphic modification, and it was the same as the started materials modification. It is my answer.
Okay, thank you, Prof. To Mbak Elok, because maybe pertanyaan ini akan dijawab selanjutnya ya, karena Prof. Piroska belum membahas sampai sedetail itu. Okay, the next question. This is from Dia Lutfia. What is the best method from crystallization, Prof? Because before you said several methods. What is the best method, Prof?
Best method for
making crystallization, or maybe for making amorphization.
The best. It's a hard question because it is influenced by the API properties, and in case of conventional crystallization, I think the very good method is a solvent method, and the pharmaceutical industry uses this method. First of all, this method with cooling. It's very simple, and if the API has very poor solubility, or the reaction is very important, it may be the reaction method, the crystallization by reactive crystallization, but also it depends on the habit or the property of the API. In case of amorphization, the method, the answer is same. The method is influenced the property or the properties of the API. In case of good solubility agent, we use solvent. First of all, we use the solvent methods. It is because we can dissolve the API in ethanol, methanol, and if the API has a good amorphous property, first of all, I suggest the solvent method.
Okay, thank you, Prof. So I will open the directly question. And there want to directly question participant? Okay, maybe this is the last question, Prof. This is from Angga Pratama. I will try to translate in easily word. Prof, why spherical crystallization is recommended, especially for high dose or active ingredient? And then what happens if we use the low dose? Maybe the importance of spheric in a high and in the low dose. There have differences, Prof?
Okay, the question is about method. can I suggest, because of low cost for crystallization and
yes, he asked about. Yes, he
Yeah,
asked
okay.
about. Okay, he asked about what is it the effect of spherical crystallization, Prof.
Mhm.
Can I use in can we use in high and low dose, especially for formulation?
Tehát, hogy a szférikus kristályosítás. Okay. Okay,
you
my
get it,
answer.
Prof? Sorry. Are you get it the question, Prof?
Yes,
Okay,
yes, I see. Okay. If we see the formulation, first of all, the spherical crystallization is suggested because we have such habit, the API, which is suitable, the prompt pressing, tablet pressing, and the cost, I think, not will be not too much or more than in case of conventional method, because we delayed the wet granulation during the tablet making, for example. And the pharmaceutical industry look for the possibility of the spherical crystallization. And nowadays, the industry has some research work to form the API to spherical form, because it is suitable for direct pressing and prepare the DC quality API. And the tablet parameters and the tablet making will be better, and we can prepare, and it's very important, I think, we can prepare the tablet with a high amount of active agent.
Okay, thank you, Prof. To Mas Angga, maybe I will translate in Indonesia. Jadi kalau untuk kristalisasi itu tergantung dari zat aktif, tidak bisa digeneralisir bahwa bentuk kristal A atau semua bentuk kristal A tidak bisa dijadikan untuk dosis dalam skala besar, ya, karena tergantung dari masing-masing zat aktifnya. Okay, Prof. Maybe we now in the closing time. Would you give our the closing statement, Prof? Would you give closing statement?
What you gave them? Yes, I would like to say again that we can have ready for contact or cooperation, and we can have a discussion by email about the topics. And I would like to say you that so I'm emerita professor in the institute, and the director of this institute is Professor Ildikó Csóka, who is an expert in QBD and the same topic. And if you choose or select the topic for cooperation, I can contact with Professor Csóka, Ildikó Csóka, and after that you can have a cooperation with the colleagues, but first of all, the director of the institute. Okay.
Okay, Prof. Thank you for giving us such informative and interesting material, Prof. Piroska. Okay.
Okay.
Okay, ladies and gentlemen, we finally come to end of this lecture. Before I close case lecture, I like to take the conclusion for what the speaker has presented. First, pharmaceutical formulation of drug substance, change of physical chemical properties, for example, manufacturing aspect, can affect particle habit, particle size, and etc. And the second, the differences of crystallization, polymorphism, amorph are below. First, crystallization is a separation process where a solid product is obtained from liquid or gas phase. The aim of this crystallization or make crystallization, first purification, change particle form, size, surface, and can be influenced of biological effect like dissolution. Many factors that influence crystal formation, such as stirring rate, and then the usage of co-solvent and the mixing process. And then the second about polymorphism, it is like crystal, but the crystal structure is different. Polymorphism screening is of great importance in preformulation testing of active ingredient. And then the next, amorphization. It is solid state like crystal, but the molecules are not packed in repeating long range and ordered fashion. The aim of amorphization of crystallization material, the important, it is increase the solution of water insoluble active and increase compressibility and a new pattern of active agent with extended. Yes. Okay, okay. I want to thank you, the speaker, especially for Professor Piroska, for the informative and exciting presentation, and all the participants for their active participation. Finally, give applause to the speaker.
Okay. Thank you very much again for the invitation, and I wait for the email to answer your questions.
Okay. Okay, Prof. Thank you so much.
Okay. Okay.
Okay, Miss Gita, are you will speak in module language?
No.
To closing this guest lecture.
Yes.
Because just only Miss Gita can speak in module, Prof.
But the Hungarian language is hard and not so simple. Okay, but I hope that in the future you can have a cooperation with my colleagues and the institute, and our institute waits for PhD students from Indonesia, and it may be the other cooperation maybe between the two faculties.
Sure. Thank you so much. Thank you so much, Professor.
Thank you so much. Thank you so much, Prof. Piroska.
Okay. Okay. Thank you very much again.
Maybe before we closing this moment, we will have photo session. Okay, open your camera.
Yes.
Yeah, we will photo session, Prof. We will have photo session
for all the participants.
Yes.
Okay.
Yes.
From slide 1, I will continue. Okay, 1, 2, 3. Okay, and then the next slide. 1, 1, 2, and 3. And then the next slide. Oh, the next slide, just only profile picture. Okay, thanks a lot, Prof. Piroska. Maybe next I will email to you to ask about postgraduate program, especially in pharmaceutical technology field, Prof.
Yeah. Okay.
Okay.
Okay. Thank you very much again.
Okay. Thank you, Prof.
See you.
Okay, maybe in closing, thank you very much for your attention. Wassalamualaikum warahmatullahi wabarakatuh. Good afternoon. Good morning to Hungary, Prof. Terima kasih semuanya atas partisipasinya. Terima kasih.